Hello histology students. This review session will cover everything that we've done from week 1 through week 4, all of the topics, descriptive text as well as Myers histology. Um, I, I emphasize that this review is meant as an aid to your studying. It does not substitute for reading the descriptive texts and studying Myers histology. I would say that about 75% of what you'll encounter on the midterm exam will come from or will have been covered in this review, but the other 25 odd percent will not be covered and will be in the descriptive texts and in Myers histology. I think this review will take 1.5 to 2 hours, so you can come back to it. I'm actually going to do it in little segments so that you can watch a segment for about 5 to 10 minutes and not have to watch, say, the whole segment in order to get to something like skin. So we're going to start with staining, which is the first topic of any consequence in this course. I did mention sometime earlier that there were only a few stains that you really needed to be able to identify for this part of the course. One of those is the elastic stain, which is very easy to recognize, stains elastin black. Our old friend hematoxylin and eosin stains nucleic acids, nuclei, ribosomes, etc. blue-purple, and pretty much all proteins, muscle mitochondria, collagen, etc. red. Masson's trichrome is one that you need to know. It's a general connective tissue stain, staining collagen, collagen blue, and then muscle erythrocytes, etc. are red. And it does stain elastin, but that's not something that you've really had a look at. But it varies from light blue to light pink. And then the other one which you should be able to recognize is the periodic acid shift stain, which stains substances that are rich in sugars, particularly basement membranes, and glycogen mucus secreting cells in some of the glands that you've looked at. And I don't think there is anything here that you need to know. Trichrome is pretty much the same as Mallory's. So let's look at a couple of these stains. Here is the periodic acid shift stain, which stains sugar-rich substances such as basement membrane, glycogen or mucus secreting cells. And as you can see here in this section taken from the kidney, the basement membranes, the basal laminas surrounding the epithelial cells, surrounding these tubes which are lined by epithelial cells, are stained very red, as is other types of connective tissue within these within the kidney, and it stains red blood cells, this yellowy orange color. Doesn't do too much for other cells surround in these tubules. The nuclei are brown, but this would not be a stain that one would use in order to show nuclei. Masson's trichrome is a stain which is used to stain connective tissue and muscle and be able to distinguish the two from one another. It stains connective tissue very blue, and all muscle. I've here brought up a slide of cardiac muscle, but the same would be true of skeletal muscle and smooth muscle. It stains it red. Where you have a mixture of skeletal muscle and cardiac muscle, it will show up purple as is shown in this area here. And additionally, it stains erythrocytes, red blood cells, in the small vessel here, a red color as well. And finally, elastic stain is a very easy one to see. One has lots of elastic in the connective tissue, particularly underlying skin, underlying blood vessels, and it stains these elastic fibers a very dark black color. This is a vein, I would imagine, and you can see that there's some elastic in the wall of the vein here. The nuclei, the endothelial cells lining this vein, are also stained black, but that's not because they have elastic in them. It's just because it's a characteristic of this stain.